Now that you've learned in general how cells communicate, today we're going to talk about signal transduction, which are the specifics of how cells actually send those messages and how cells kind of know how to respond to their environment. So the general overview here of signal transduction is, a cell needs to send and receive messages. Either they're getting All right, so now we've learned a little bit in general of some different ways that cells can communicate. But now today we're going to talk about signal transduction, which are the real specifics of how those messages actually get delivered to the cell. Okay, so the whole idea here is through signal transduction, we are allowing messages to go in between cells. We're also allowing cells to respond to the environment. And oftentimes, if you're looking at this diagram here, this activation of cellular responses, lots of different things can happen, but one of the most often things is that we're like turning on or turning off a gene, which is going to then affect protein production. Okay, we haven't gotten into this yet. We'll get into it later in the year, but the whole thing with genes and chromosomes is that they give instructions for making proteins, right? Okay, so that is oftentimes the response that the cell is having. Okay, so with signal transduction, we have three general steps here. The first step is the reception step. So this is where the cell just actually gets the signal. And we're going to go through each one of these in more depth in just a second. And then the transduction of signal transduction is the message getting transferred inside of the cytoplasm. Okay. And then the third step is the response. What is the cell going to do because it's gotten this message, right? And so we just talked about how oftentimes it's a gene turning off, getting turned off or turned on. It can also be just some kind of different cellular process that's activated. Could be apoptosis. It could be certain things develop or proliferate, any number of things. Okay. So again, we're going to go into each one of these three more in detail right now. All right, so here we go. The cell receives the message. This is happening in the cell membrane. Okay, so here's the membrane, and here's our receptor protein. It's an integral protein. And so we're going to get a message, possibly from another cell. And this message is coming in the form of a chemical messenger, which is called a ligand. And we learned about these in our short distance and long distance communication from the last lecture. Okay, so these could be things like hormones, neurotransmitters. Okay, and so this hormone or neurotransmitter is going to be in the fluids, possibly either in the inner space between two cells or coming through the blood. Okay, and then it's going to come and find this receptor. Notice that the shape, similar to enzymes, this shape ligand is going to match exactly this shape receptor. Okay, so it's similar to enzymes and how we have that shape function match up, right? So here we are again, only this ligand will go into this receptor. Okay, and if you're looking in your book, there's lots of different kinds of names for these receptors. There's a ligand-gated ion channel. There's a G protein couple receptors. You don't really need to like memorize these, but just so you know, if you see these words later, there's RTKs. These are all just different names of different kinds of receptors. Okay, and so basically, once this ligand comes in here and binds the receptor, this receptor is going to change its shape. So maybe this oval shape would change ever so slightly, okay? And when this thing changes its shape, this is going to pass the message to the inside of the cell. So notice that this ligand never enters the cell. It just causes a shape change of this integral protein so that this inner part of the protein is going to do a shape change, and that's going to kick off a whole series of reactions inside the cell, which is transduction. Okay, so here we go. Here's our receptor protein. Okay, it changes shape. And then there's going to be some relay proteins. And so basically what's going to happen is it's going to be like a domino chain reaction kind of effect where one is going to activate the other, is going to activate the other. These things can be called secondary messengers, okay? And so this is going to transmit this message from outside to the inside and probably end up in the nucleus, okay? So here they're getting passed from protein to protein to protein, okay? And oftentimes the signal gets amplified. Okay, and so we're going to talk about that in just a second through a phosphorylation cascade. What? It's okay, hang with me. So basically, when our ligand changes shape, this is going to activate, kind of turn on this molecule. Okay, and so now this guy is active. Okay, and then we're going to use up some ATP, and then the P, one of these phosphate groups, now is going to come on here. So this guy is now activated. Great. And we're going to use another ATP, and then this guy gets activated. We're going to use another ATP, and this guy gets activated. Okay. So do you see how like one protein gets phosphorylated, and then that phosphorylates something else, and that phosphorylates something else, that phosphorylates something else, kind of like a domino or waterfall or cascade effect. Okay. And that is going to end up in the next step giving us some kind of response. Okay, so this is the transduction, is this message getting passed from thing to thing to thing to thing to thing to thing to thing. And at the end here, we get our response from our cell. Okay, typically this is something's happening in the nucleus. We can turn on a gene, turn off a gene. This could start a protein being made, or it could stop a protein from being made. It could also be a different kind of thing, like, hey, let's open this channel or close this channel, or let's change our metabolic rate. It can be any one of those things, but it often involves proteins being made or stop being made. Now, if one of these proteins in this whole thing had a mutation, what do you think would happen? Well, if it's not quite formed right, there's a chance that this cascade might not actually happen. And so the signal that your cell is getting might not get all the way to the nucleus. So the signal would stop. So the message wouldn't get delivered. That would be no good. Okay. So you get one ligand binding. And so the question is like, well, how much of a response does it get? Well, as it turns out, one ligand can have a huge effect on a cell. So just this one ligand can all of a sudden produce like 3 secondary messengers, and then that one can like produce, what is it, 9, and those each can produce a couple. And so basically the idea here is your cell can get one message and turn it into a huge message to the nucleus, so you can get lots of response right away, which is really, really great for efficiency. Okay, and so one of these amplification molecules that is really, really common is called CAMP, or cyclic AMP. This is one of these secondary messengers that helps deliver the message from the membrane protein here to the nucleus. And it's just what I showed you on the last slide, okay? It's exactly this, but it's just a specific one that does this, okay? So this CAMP gets phosphorylated, and that phosphorylates a thing, and that phosphorylates a whole bunch of things, and they phosphorylate a whole bunch of things, and so on and so on and so on, until look at all the stuff that's made off of just this one signaling molecule. And actually, as it turns out, it's even bigger than that, okay? With CAMP, from one receptor and one ligand, at the end of this cascade of the phosphorylating and phosphorylating and phosphorylating and phosphorylating, you get 1 million products produced from one messenger molecule, which is huge, right? It's a massive response. So your cell can respond hugely and quickly because of this, okay, which is a nice way to respond to your environment if you needed to make some quick changes for survival. So just one thing to see here. So this is an example of a really simplified example of signal transduction with deetoliation, which is greening of like plants and leaves, like when leaves start to turn green. So like light is what ends up changing the shape of this thing, which activates some secondary messengers, which activates some proteins, a kinase, and that's going to go and cause the nucleus to do a thing. And we got some ions who are also doing a thing, which is going to cause a thing. And this whole thing causes greening. Okay, this is just like an example. And actually, it's a very simplified example because this is actually the greening process. You don't have to know this at all, but just understand that even the like cartoons that I've shown you on here for signal transduction, they're very, very simplified. And oftentimes there's lots of ligands that are coming and sending lots of different messages, and your cell is doing them all at the same time, not just one. But sometimes all of those things produce one effect, like greening. Here is apoptosis. Here's all of the signal pathways that happen for cell death, right? And again, you don't need to memorize any of this, but I really like showing this one because it shows how complicated it is. And also there's some really great names. Like look at this membrane protein. It's called frizzled. And then that leads to a thing called disheveled. The biologists have a good sense of humor. Look, this is the hedgehog protein. This is patched, right? How silly. There's a thing called death factors that can come in. Again, you don't need to know this, but just showing that there's lots of things going on that can produce lots of complex results from your cells, okay? But the overall idea is you get a ligand, you get some kind of chemical messenger, either from the environment or from another cell. Oh, just ignore that. Your receptors are going to change shape, and then it's going to pass that signal on through secondary messengers, and a phosphorylation cascade is going to amplify to get some really, really big responses. That responses could come from your cytoplasm, or they could come from the nucleus. Here are the main points of this lecture. Hopefully you can answer each one of these questions. And lastly, if you want to know where we are in the book, we're in chapter 5.6.